Hello everyone, welcome to the new session of cell cycle and cell division. So this is the second part, and in this part we are going to study about the M phase, right? In the first part we have studied about the cell cycle, right? In the first session we have studied about the cell cycle. In this part we are going to study about M phase, right? So let us start this session. Though, for convenience, mitosis has been divided into 4 stages of the nuclear division, like that is the karyokinesis. Okay, and it is very essential to understand that cell division is a progressive process and a very clear-cut lines cannot be drawn between the various stages. So we are having the cell division. Cell division is having two important steps. That is, first one is karyokinesis, right? That is the division of nucleus. So nucleus divides into 2 in this stage, right? Next one is the cytokinesis. In this, division of cytoplasm occur. After the division of nucleus, division of cytoplasm also occur, right? So the two important steps. Now this karyokinesis is again, can be divided into 4 different steps. That is prophase, metaphase, third anaphase, and fourth one is the telophase, right? So what it is saying, that there is a progressive process and very clear-cut line. So we are having a clear-cut line between the prophase and metaphase stage, a clear-cut line between the metaphase and anaphase stage, a clear-cut line between the anaphase and the telophase stage. So what does this clear-cut line means? That there are different processes and different steps occur in the prophase, and that is fixed. It is not going to overlap with the metaphase in this type of cell, and it can be seen, this particular steps can be seen in the metaphase stage in the another cells. No, it never happens. So the stages which occur in prophase is fixed. So that is why there is a clear-cut demarcation between the prophase and metaphase. In the same way, the stages or the processes that occur in the metaphase stage, it is fixed, and it can never be seen in the anaphase stage, right? So there is a clear-cut demarcation, right? Clear-cut lines that cannot be drawn between the various stages. So the karyokinesis involves the following 4 stages. Just now we discussed that the karyokinesis is having the 4 stages. We can study the karyokinesis process in the 4 stages. That is the prophase, metaphase, anaphase, and telophase. So we are going to discuss one by one these stages and see what essential steps occur in these phases for the process of karyokinesis or the division of nucleus. Okay. Yeah, so the first one is the prophase, first stage. The prophase is the first stage of the karyokinesis of the mitosis, follows the S and G2 phase. So we are having G1 phase, S phase, and G2 phase, right? So these phase is the part of interphase. So as cell get to the M phase, this is the M phase. So the first step here is the prophase. So we can see here, this prophase is followed by S phase, and then comes the G2 phase, and then comes the prophase, right? So S and G2 phase In the S and G2 phase, the new DNA molecule formed are now distinct but intertwined. So, in the S phase, the duplication of the DNA occur, that is intertwined, like this. It is intertwined. It is not separated. So, it is intertwined like this. So the prophase is marked by the initiation of the condensation of the chromosomal material. So the first thing occur in the prophase is condensation of the chromosomal material. The condense means the nucleus this DNA get intertwined, and the condensation of chromosome material can be seen here. Right, the chromosomal material becomes untangled, okay, during the process of chromatin condensation. Means the one which was tightly intertwined now become loosened or untangled, like this, untangled. So this is called as the chromatin condensation. Okay, the centrosome which had undergone the duplication during the S phase of the interphase now begin to move towards the opposite poles of the cell. So we in the interphase we have discussed about that the two thing duplication occur. Okay, the first one is the duplication of the DNA, right, that occur inside the nucleus. But there is also the duplication of the centrosome which occur outside the nucleus, that is in the cytoplasm, right? Now what happened in this prophase? This centrosome, okay, this centrosome, duplicated centrosome, is moving towards the pole. It moves towards the pole. Right? So the completion of the prophase can thus be marked by the following characteristic event. So what are the important events that occur in the prophase? So the chromosomal material condenses to form compact mitotic chromosomes. Okay, so we can see the condensation of the chromatin material, and the compact mitotic chromosome you can see. Right, chromosomes are seen to be composed of the two chromatids attached together at the centromere. So at the centromere we can see that the chromosomes are attached, the chromatids of the chromosomes are attached at the centromere, and the nuclear envelope degenerates. So in the late prophase, the nuclear envelopes degenerates. The centrosome which had undergone duplication during the interphase begins to move toward the opposite poles of the cell. This we have already discussed, that the centrosome which has been synthesized, duplicated in the interphase stage moves towards the pole. Each centrosome radiates out microtubule called the ester. So we can see different microtubules around the centrosome. So that is called as esters. The two esters together with the spindle fibers form the mitotic apparatus. So mitotic apparatus takes place in the prophase stage only. Cells at the end of the prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus, and the nuclear envelope. So most of the cell organelles, right, they get degenerated along with the nucleus and the nuclear envelope. So all the content of the nucleus now you can see in the cytoplasm only, because the nuclear envelope gets degenerated. Okay, now comes the second stage, that is the metaphase. The complete disintegration of the nuclear envelope marks the stage of the second phase of the mitosis. In this particular stage, there is complete disintegration of the nuclear envelope or the nuclear membrane takes place. It started in the late prophase, but it's completed in the metaphase stage. The chromosomes are spread through the cytoplasm of the cell. Now, this chromosomes you can see in the cytoplasm because there is a no nuclear membrane. So, all the chromosomes are present where? They are present in the cytoplasm itself. By this stage, the condensation of the chromosome is completed and they can be observed clearly under the microscope. So this is the perfect stage to see the chromosome, because the maximum condensation which has been started in the prophase stage, but it's completed in the metaphase. So at this particular stage, the condensation of the chromosome gets completed, and it is the well stage, or this is the best stage to see the chromosome under the microscope. Right? So this is then stage at which the morphology of the chromosome most easily studied. You can see the appearance of the chromosome, okay, the size of the chromosome. So that is called as the morphology of the chromosome, can be very well studied at this particular stage, because at this stage the chromosome can be seen clearly. At this stage, the metaphase chromosome is made up of two sister chromatids. Now, the chromosome which was like this, earlier like this, right? It separate out in two chromatids like this. So, these are the chromatids. Okay, so at this stage, the metaphase chromosome made up of the two sister chromatids which are held together by the centromere. So here we can see that they are held together by the centromere, right? Small disc-shaped structures at the surface of the centromeres are called kinetochores, can be seen. So here you can see a disc-shaped structure, both the side disc-shaped structure, that is called as the kinetochore. Here the disc-shaped structure, that is the kinetochore, are present, right? Okay, so kinetochore serves as the site of the attachment of the spindle fibers to the chromosome. So we can see the kinetochores like this. This is the chromatids. This is the centromere, right? And this is the small kinetochores, like kinetochores. Now the spindle fibers get attached to this kinetochores. Okay, so metaphase is characterized by all the chromosome coming to lie at the equator. So if this is a cell and these are the chromosomes, they are arranged at the equator. And this is the kinetochore. And here we can see through the pole, spindle fibers are attached to the kinetochores like this. Okay, so this is the equator region. This is the equator. Okay, with one chromatids of each chromosome connected by its kinetochore to the spindle fibers from one pole, and its sister chromatid connected by its kinetochore to the spindle fiber from the opposite pole. So here we can see one of the chromatid kinetochore is attached to the spindle fiber from one particular pole, and another chromatid of the same chromosome is attached with the another pole with the help of spindle fibers, right? So this type of arrangement can be seen in the metaphase stage. The plane of the alignment of the chromosome at the metaphase is referred to as the metaphase plate. So like this, the alignment of the chromosome at the equatorial region or the equator things, so that is called as the metaphase plate. The key features of the metaphase are spindle fibers attached to the kinetochores of the chromosome. So this spindle fibers, very important, get attached to the kinetochores of the chromosome. Okay, chromosomes are moved to the spindle equators and get aligned along the metaphase plate. So this is very important. Metaphase plate means arranging all the chromosome at the equator, through the spindle fibers movement. Okay, so this is very important stage. The third stage, that is the anaphase. So at anaphase, each chromosome arranged at the metaphase plate is split simultaneously and the two daughter chromatids. Now, what happened? We have seen that this chromatids of each chromosome is attached with the spindle fibers of the running opposite poles, right? In this way. But what happened in the anaphase? This gets split. And the movement of this chromatid with the help of this spindle fiber toward the opposite poles, so these are referred to as the daughter chromosomes. So these are referred to as the daughter chromosomes of the future daughter nuclei. Okay, begin their migration towards the two opposite poles. So we have seen this equatorial plane or metaphase plate at this stage, but after this stage in the anaphase, when the cell get entered in the anaphase, this spindle fibers makes its movement in such a way that the chromatids of the chromosome get separated. The chromatids of the chromosome get separated in this way and start moving towards the opposite pole. Okay. So here you can see, this is the movement of the chromatids with the help of spindle fibers. The chromosome moves away from the equatorial plate. Now here you can see no equatorial plate like structure. Okay, so the centromere of each chromosome remains directed toward the pole. So here you can see this is the centromere. And it is directed where? Towards the pole. This is the pole. It is directed toward the pole. Okay, and hence at the leading edge with the arms of the chromosomes toward trailing behind. So here you can see these are the arms of the chromosome behind, but this centromere of this chromosome is towards the pole, because it is attached with the spindle fibers, and the spindle fibers just keep on moving towards the pole. Thus, anaphase stage is characterized by the following key events. Centromere split and the chromatids get separated. Chromatids move toward the opposite poles. So this two stages, these two things occur in the anaphase. Okay. Next comes the telophase. At this stage, the chromosomes that have reached their respective poles decondense and lose their individuality, right? So at the starting of the prophase. There is a lot of condensation of the chromatin material, but here decondensation process, just opposite of that, occur at the telophase stage. Okay, the individual chromosome can no longer be seen, and each set of chromatin material tends to collect at each of the two poles. So we are having two set of chromatin material, one at one pole and another at another pole, like this. Okay, this is the stage which shows the following key events. The chromosome cluster at opposite spindle poles and their identity is lost as a discrete element. So chromosome identity is lost at this particular stage. The chromosomes can never be seen in this stage, right? So nuclear envelope develops around the chromosome cluster at each pole, forming the two daughter nuclei. So the cell, one individual cell, is having two poles, and each pole is having two daughter nuclei, two daughter nuclei, right? So this, at this stage, this two daughter nuclei is formed. Nucleolus, Golgi complex, and ER reform. Okay, so these organelles can be seen again in the cell now. Now, after this karyokinesis, so till here we have studied about the karyokinesis, in which what happened? Prophase, metaphase, anaphase, and telophase. These are the four different stages in which the division of the nucleus occur and the two nucleus is formed in one cell. Now, the cytoplasm divides. Okay, so that is the cytokinesis part. After the karyokinesis, the cell itself is divided into two daughter cell by the separation of cytoplasm called cytokinesis, right? So at the end of which cell division gets completed. Now, after the karyokinesis, the part is left is that is cytokinesis, in which the division of cytoplasm takes place, and this is the end of the cell division. In animal cell, this is achieved by the appearance of furrow in the plasma membrane. So furrow-like things occur in the plasma membrane. So here we can see a furrow or cleavage, right? So the furrow gradually deepens. So it deepens and ultimately joins the center, dividing the cell cytoplasm into two, and it forms a two individual cell. In plant cell, they are enclosed by the relatively cell wall. Therefore, they undergo cytokinesis by the different mechanism, because the plant cell is having cell wall also along with the cell membrane. So their division of the cell or the cytokinesis is quite different. In plant cell, wall formation start in the center of the cell. Okay, like this. It forms. So these are the two nuclei. It forms at the center of the cell like this, and grows outward to meet the existing lateral walls, and it grows outward to meet the lateral existing walls. The formation of new cell wall begins with the formation of simple precursor called the cell plate. So cell plate formation takes place in plant cell that represent the middle lamella between the walls of the two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastid get distributed between the two daughter cells. Okay, so after the cytoplasmic divisions, these organelles like mitochondria, plastid, okay, they get distributed between the two daughter cells. In some organism, karyokinesis is not followed by the cytokinesis, as a result in which multinucleate condition arises, leading to the formation of syncytium. So what happened in a cell? the nucleus divides into two, and this nucleus again divide, and lots of nucleus you can see in a single cell. So that is the syncytium. No cell division is taking place. And example is the liquid endosperm in the case of coconut. Okay, so what is the significance of mitosis now? Mitosis or the equational division is usually restricted to the diploid cells only. So this equational division or the mitosis is a significance can be seen significantly in the diploid cells only. However, in some lower plants, in some social insects, okay, lower plants like in Chlamydomonas pyrogyra, social insects like honeybee and ants, haploid cells also divide by the mitosis. It is very essential to understand the significance of this division in the life of an organism. The growth of multicellular organism is due to the mitosis. So we have already discussed that the division in the body cells or the somatic cells occur by the process of mitosis. So multicellular organism, the growth is done by mitosis division. The cell growth result in disturbing the ratio between the nucleus and the cytoplasm. This is essential for cell to divide to restore the nucleocytoplasmic ratio. So this is the important division in which the nucleocytoplasmic ratio division takes place, right? A very significant contribution of the mitosis is the cell repair. So when there is a when there is a requirement of the repair in the cell, okay, so this type of division takes place, that is the mitosis, right? So mitosis usually result in the production of the diploid daughter cell with identical genetic complement. So when there is a mitosis division, we are having a identical diploid cell or the diploid daughter cell. They are identical genetic complement. Their genetic material are identical, right? The cells of the upper layer of the epidermis, cells of the lining of the gut, and the blood cell being constantly replaced means new cells are formed. These cells are constantly replaced by forming the new cells. And how these new cells are formed? By the process of mitosis, right? Mitotic division in the meristematic tissue, the apical and the lateral cambium result in a continuous growth of the plant throughout their life. So this is very important for the plants. Like in the plant, they are having a continuous division of these cells, continuous division, like we can see in the formation of the lateral cambium or the meristematic tissue, right? So this occurs due to the process of mitosis, by the process of mitosis. So this is all about this session. We have completed the mitosis division. We will see one more division, that is the meiosis, in one more session. Till then, thank you very much.